Let's look at our first question. I'm going to skip this. I'm going to come back to this one. I wanted to do this because I thought this was good introduction. This question here is asking us to define alleles and recessive. So I thought we could make some notes on that first. So let's write some definitions. First of all, let's start off with definition for a gene. What's the definition for a gene, guys? Until 8:45, yeah, that's when it finishes. Genetic information, a bit, section of DNA, yes. Section of DNA that, there we go, Georgia, thank you. Codes for a specific protein, yes. Section of DNA that codes for a particular protein. So you've got a really long strand of DNA, which is just a code for your whole body. So imagine like a really long strand. A gene is one small section of it. So the whole strand codes for everything in your body, but one small section we call a gene. That small section might code for like proteins in your skin, or it might, and another one, so this section here might code for enzymes, like enzymes in your stomach or something like that. A gene is just one small section that codes for one particular protein. Okay. Next, let's define an allele. What's the definition of an allele? Yes, different version of the same gene. So different version of the same gene. So we'll say, yeah. So what do I mean by that? How could it be different and same at the same time? So when I say same gene, I mean like eye color. That's a type of gene. Different versions of eye color could be brown eyes, blue eyes, green eyes. So those are different alleles because they're the same gene, eye color gene, but different versions of it. That's what it means. Next, let's define. Oh, another thing about alleles, they always come in pairs. So they always come in twos. And that's when you come across dominant and recessive. What are the definitions of these? Not same type of gene, no. Allele that needs two copies to be expressed, yes. Dominant appears the most, kind of. One copy is required. That's not going to give you enough for a mark. Always expressed, yeah, good. So dominant, so remember I said they always come in pairs. So you can have, so dominant's always represented with a capital letter. So it might be like a capital R, capital D. Recessive is always represented with a small letter. And they always come in pairs. So it would be something like capital A, small a, for example. Or it could be small a, that just means two recessives. So dominant, if a dominant is present in your pair, it's always expressed. So whatever characteristic it shows, so if, like I said, if it was eye color, if it was brown eye color, and it's present, if a dominant allele is present, that means that person's going to have brown eyes. Even if their other one is, if they have one brown-eyed allele and one blue-eyed allele, the dominant one will always take over. So always expressed. Recessive, on the other hand, only expressed, I think people already wrote it down, only expressed when both alleles are recessive. So here it wouldn't be expressed because there's a dominant there, but here we got both as a recessive, so it'll be expressed here. Next, we got two other terms. We got homozygous and heterozygous. What do they mean? Yeah, homozygous means both alleles. Remember, I said they come in pairs. When both alleles are the same, is homozygous. And heterozygous is when they're different. Heterozygous when both alleles are different. Good. Quite a lot of definitions today. Another two, we've got genotype and phenotype. Anyone know those? It's taken up the whole page. Yeah. Good. Phenotype genotype is literally the two alleles, the pair of alleles you have. So if they ask you what is the genotype, you would give an answer like capital A, or small a, or capital A, capital A. That's the genotype. It's literally the two alleles that you have, the combination of alleles that you have. Yep, so example, capital A, small a, capital A, capital A, small a, small a. Phenotype is the physical characteristics. You can remember that because it begins with PH, physical. It's what it actually physically does. So brown eyes, blue eyes, brown hair, any physical characteristics is the phenotype. So it's what it comes out of the two genotypes. Yeah, so e.g. brown eyes, blue eyes. Okay. That's a good start, I think. Now let's look into the question. So we got two definitions here. Alleles, recessive, we've defined them both. So literally write down what we wrote. So alleles, different forms of the same gene. And then recessive, only expressed if both are recessive. So let's read the question now. Good idea to do that. People with cystic fibrosis, that's a genetic disorder, make large amounts of thick, sticky mucus in their lungs. Cystic fibrosis is caused by inheritance of recessive alleles, okay, not dominant ones. So they're only expressed if both are recessive. Mr. and Mrs. Brown have a child with cystic fibrosis. They hope to have another child. They want to know the probability that their next child will have cystic fibrosis. They visit a genetic counselor who explains you're both heterozygous for cystic fibrosis. There's a 1 in 4 chance that your next child will have cystic fibrosis. So here we go. An allele for a person being affected by cystic fibrosis. Sorry, unaffected and affected. So someone who has cystic fibrosis. So it's recessive. It said it's recessive here. That's why they use a small n here. That means a person who doesn't have it is going to be dominant with a capital N. So Mr. and Mrs. Brown both have the same genotype. What is their genotype? They're heterozygous, literally says they're heterozygous. Heterozygous means different alleles. So it's going to be capital N. Yep, well done guys. Capital N, small n. Okay, next one is a genetic diagram question. 1 in 4 chance that Mr. and Mrs. Brown's next child will have cystic fibrosis. Use a genetic diagram to explain why. So genetic diagram. You can draw a Punnett square. So we've got Miss Brown here, his alleles, and Mrs. Brown here. So in a Punnett square, you write down the genotypes here and here. So we said they're both heterozygous, they're both capital N, small n. So capital N, small n here. And then Mrs. Brown's the same, capital N, small n. And then what you do is you combine them in the squares in the middle. So here, that's a combination of capital N, capital N. Here is a capital N, small n. Here is a capital N, small n as well. And then here we got 2 small n's here. 1 in 4 chance that their next child will have cystic fibrosis. Remember, cystic fibrosis is a recessive allele. So they only have cystic fibrosis if both are recessive. So this is the only combination that the person's going to have cystic fibrosis. These two will not be, because it's recessive, and the dominant allele, which doesn't have cystic fibrosis, that's going to take over. So we point to that and we say 1 in 4 chance or 25% chance of having cystic fibrosis. Okay. Does anyone have any question on this? Any questions? Is the one with the dominant allele a carrier? Yeah, they're both carriers. So what a carrier is someone who has the allele but doesn't have the disease. So Mr. and Mrs. Brown don't have cystic fibrosis, but they have the allele for it. So they're carriers. So that means their child has a chance of getting it. You can't be a carrier if the disease is dominant. Because if you have the dominant gene, that means you have the disease. So there's no way you can have the gene and not have the disease. So it only comes if the disease is recessive. Does that make sense? Do you need to name the people? No, you don't need to write Mr. Brown, Mr. Brown. You don't need to write any of that. You can go straight into the diagram doing that. Okay, good. Do you always put capital first? No, it doesn't matter. You could do it the other way around. Doesn't matter. It can be small n, big N. It can be the other way around. You'll still get the same result every time. As long as you get the two combinations right, you'll still get the same result. What happens if the disease is dominant? Okay, if it was dominant, let's think about that. If it was dominant, guys, what would the percentage be? If, let's say the disease was capital N, what would the percentage be? It would be 75%. Because if it was dominant, if it was dominant, it means anything with a capital N would have the disease. So that would include this and this, as well as this. So there'll be 3 in 4 chance. This is the only combination where the person wouldn't have the disease. Okay? If it's the small n and big N combined, does it always have to be the bigger in front? Not necessarily. Usually it is written like that, but I don't think you'll lose marks if you do the smaller one in front. But I'd say I always see it in textbooks. I always see the capital one first. So try and write it like that, but I don't think you'll lose marks if you don't. Okay, back to this question now. So this is about selective breeding, this question. Many different types of animals are produced using selective breeding. Some cats are selectively bred, so they do not cause allergies in people. So there's 2 other reasons why people might selectively breed cats. What do you guys think? Yeah. Looks like fur, yes. Anything like aesthetic, you'll get marks for saying anything aesthetic. So like better looks or more fur, less fur. So we'll say less fur as an example, but anything aesthetic will get your mark. What else do you want from a pet generally? Gentle nature. Yeah, you want them not to be aggressive. That's a common one I see on mark schemes all the time. Gentle nature or not aggressive. It's in the textbooks as well. So these are the ones you go for, but you can say other stuff. Yeah, more domestic is fine. What does that even mean? You want them to have a calm nature. You want the animals not to like bite, things like that. Because some dogs bite more than others, isn't it? Just saying aesthetic is fine as well. Less fur could be less fur. It's just an example. Some animals, they might want less fur. Some animals you might have more fur. It's different reasons. Behavior, yeah, I'd say behavior as well. Say better behavior. Don't just put behavior. Say better behavior. Can you say desired appearance? Yeah, if you say appearance, don't say desired characteristic because it's asking you for like examples. Reduces gene pool, that's not a reason to do it though. So that wouldn't be an answer. It does decrease gene pool, but that's not something you want to happen. So you can't put that. Disease resistance is fine. Yes, it is. Less genetic variation. That again, that's not something you do selective breeding for. So that wouldn't be an answer. No. Reduce the passing on of illnesses. No. Reduce the chance of having an illness, yes, but it doesn't reduce the passing on of illnesses. It just reduces the chance of that animal having that disease. What's gene pool? Gene pool is if you had like 10 cats in your stock that you wanted to selectively breed, if they were completely unrelated to each other, then they'll all have like a really good variation of genes, right? They'll all be, imagine 10 different people from all around the world in a room. Yeah. But then if you got ones which were closely related, so like brothers, sisters, cousins, then their genes are more closely related as well, right? So that means there's a reduced gene pool because there's a smaller variation of genes within all the individuals. So selective breeding causes a reduced gene pool because what you do is you pick the two best individuals from the stock, and then you breed them, right? So you have your two best ones and then you breed them, and then they give offspring. So they might give like 7 offspring, but then what they do inbreeding after that in selective breeding. Then out of the offspring, they pick the two best ones. So maybe this and this, and then they breed those two. So what's going to happen is all the stock, as the further down the generations you go, all the different animals, they're going to be really closely related. So they're going to have really similar genes. And if that happens, then that's bad, which we'll talk about in a second. That's actually the next question. It says selective breeding could cause problems of inbreeding in cats. One problem of inbreeding, what would that be? What would one mark for that be? Why is it bad to inbreed? Yes, they're not more risk of mutation. It can lead to disorders. It can lead to recessive disorders like cystic fibrosis. The reason why is if they're closely related, that means they're more likely to have recessive genes. They're more likely to be similar in genes. So they might be carrying the same recessive genes. And these are more likely to get passed down if they're closely related, because this one would have the recessive gene and the other one would have the recessive gene as well. If they both have the same recessive gene, there's a chance that this disease, whatever it is, would get passed down. But if you breed this cat with something that's completely unrelated, so it's not in this gene pool, if it's a completely different cat from outside, it's more likely to have this combination. It's more likely not to have a particular disease. So they'll be dominant. So they're less likely that if these two bred, there's no chance, there's like a 0% chance of their offspring getting a disease. I hope that made sense. They're more likely to pass on, we'll say recessive disorders. Although you don't need to say that for your answer. Less genetic variation, it does, but it's basically, why is that bad? Less variation, why is it bad? Because of this. That's why. Can we say they won't have a survival advantage? No, not really. That doesn't really answer the question, because that wouldn't necessarily lead to that. Can you say reduces gene pool? Not for this one, no, because it's basically asking what would the reduced gene pool lead to? Why is a reduced gene pool bad? So it's asking you for one step further in this question. Physical deformity? No, not necessarily. A recessive disorder could cause a physical deformity, yes, but physical deformity could come from just a normal mutation as well. So I wouldn't say that. Inherited defects, yes, you can say that. That's fine, because that's like an inherited disorder, isn't it? Okay. Let's move on. Many people have breathing problems because they're allergic to cats. The allergy is caused by a chemical called FeLD1. different cats produce different amounts of FeLD1. A cat has been bred so that it doesn't produce FeLD1. The cat does not cause an allergic reaction. Explain how the cat has been produced using selective breeding. Okay, it's basically just asking you the steps for selective breeding. There's 4 steps for 4 marks. 4 steps. What do you guys think? Male cats produce more FeLD1 than female cats. That's, I don't know, is that a fact that you know? You can't tell that from the question. Yep, they select they select 2 cats with desired characteristics. Yep, that's the first step. Good. Choose 2 cats with we'll say what the desired characteristic is in this case, because I think they want you to use the actual example. So 2 cats which produce low amounts of FeLD1. Let's relate it to the question. Then breed them. Yep. And excellent. So after that is what I was explaining here with this diagram here. So after that, you get out of the offspring, you pick the 2 best offspring. So choose 2 from offspring with the best characteristics. And then final step is just repeat the process for several generations. So saying it will survive and reproduce, that's more of an evolution answer. There's no need to, it's not about them surviving because they're pets, right? So someone's looking after them. It's not like they're in the wild or anything. So you don't say that. So repeat the process for several generations. Yeah, until they all have the desired characteristics. So what's going to happen is, as you do this more and more, as you go on with more generations, you're going to get more cats that produce less of this FeLD1. So that's a good thing. So that'll be a good thing for pet owners, basically. Okay, does that make sense? Any questions on that? Yes. Okay, amazing. Let's move on. I think next question is not about DNA. So we got a question about hormones. So yeah, this is a bit of the menstrual cycle as well. Name glands A and B. So look at the location here. What do you guys think? We've got 1 here in the brain, and then we got 2 down here. Pituitary gland and ovaries. Excellent. Gland A produces FSH. FSH controls changes in gland B. How does FSH move from gland A to gland B? Yeah, in the blood. Or in the bloodstream. Very good. Let's move on. A woman is not able to become pregnant. The woman does not produce mature eggs. The woman decides to have IVF treatment. Which 2 hormones help the woman to produce and release mature eggs? Not the second one. It's the first one. Let me write the primary uses of each one. I think on one of the other YouTube videos, the one on hormones, I think I write these down properly. But let me quickly write the main functions. FSH, LH, estrogen, and progesterone. Those are the three that are involved in the menstrual cycle. FSH, follicle stimulating hormone, that matures the egg. LH releases the egg. Estrogen causes the lining to thicken. And progesterone maintains the thickness of the lining. They have secondary uses as well. I've got a TikTok video, it's pinned to the top. It covers all of these and the whole cycle. So look at that. It's got all the information that you need to know for that. So yeah, which one produces and releases? So the one that produces is FSH, the one that releases is LH, so it's the top one. Estrogen doesn't do that. Okay, next one. Giving these hormones to the woman helps her produce several mature eggs. Doctors collect the mature eggs from the woman in an operation. Describe how the mature eggs are used in IVF treatment so that woman may become pregnant. So it's asking for the process of IVF. Fertilizing a petri dish is a bit close. Take out the embryo. No, they don't take out the embryo from the woman. Take the egg and fertilize it. So give her LH and FSH to release the eggs. Yep, that's already been done. They've already said that here. So after that, you remove the egg and remove sperm and fertilize. So fertilize. Okay, next thing, what do you do after it's been fertilized? Then you collect the eggs. So you need to let it divide a bit. Yes, I'll put it up on YouTube. What's progesterone? Progesterone is produced in the ovaries, and it causes the lining of the uterus to be maintained. So once it's thick, so the estrogen thickens it, and then the progesterone keeps it thick until day 28. So yeah, you wait for it to form an embryo, which is basically like a ball of cells, and then you put it back into the woman. So it forms embryo, which is a ball of cells, and then inserted into the woman. We'll say the mother's womb or uterus. Okay, next one. IVF clinics have been set a target to reduce multiple births. So that's like having twins, triplets, more than that as well. at least 78% of IVF treatment should result in single babies, and a maximum of 24% of treatment should result in multiple births. So just one reasons why the clinics have set this target to reduce multiple births. So why would they want to reduce the birth of twins and triplets? And sometimes you get more. Yeah, they can lead to miscarriages, stillbirths. There's going to be risk to the mother, risk to the baby. All of that is acceptable. Can lead to miscarriage, yeah. Good. So now we've got a data question. 2 clinics, R and S, used IVF treatment on women in 2007. Doctors at each clinic used the results in the treatment to predict the success rate. Compare the success rates of the 2 clinics. So how do we work out the success rates here, or rough success rate? What paper is this? Not sure. These are loads of different questions from different papers. It's a mix from loads of different paper too. Yeah. The mean, you can't work out the mean really from this data. So remember the success rate. So how successful the IVF is. So the success of IVF basically means the woman gets pregnant in the end, right? So it's asking you to compare the success of this clinic and this clinic. So it's a calculation question. You just compare. What do you compare though? Which numbers do you compare? The range, not really the range, because the range isn't going to tell you the success rate. The range is just going to tell you the amount, the difference in the highest value and the lowest value of success. So that doesn't tell you. Yes, good. 200 and the 20. But using these 2 numbers isn't representative. You can't just say 200 is bigger than 20, so this one's more successful, right? What's a better way? What's a better way of doing it? Percentage, yeah. Looking at the percentage. What percentage of successes are there in clinic R? What percentage do we have in clinic S? So what would you do? So for this one is basically 200 over 1004, right? So which you can estimate. Yeah, 200 over 1000 is roughly 20%. Then we got 20 over 98, which is also similar to 20%, isn't it? 20 over 98. If you work those both out, they're very close to 20%. So you can say in your answer, it's not exactly 20%, guys. You can give the actual values. Can someone work out the actual values and tell me what they are for the first one? What's the value for the first one? 200 over 1004. All this for 1 mark. Yeah, it's a lot. You don't need to do this. You can just, in your head, you can work out this around 20%, this around 20%, and you can just say they're similar. 19.2 and 19.9. Is that what we got? I'm not sure. I don't have a calculator on me, guys, but let's say it's those 2. You can just say they're similar. Similar success at around 20%. That's enough to get you the mark. You don't even need to do this working out here because it's 1 mark. The range of predicted success rate in 2008 is much smaller than the range of predicted success rate for Clinic S. Suggest why. Anyone know? So remember the range is the highest value and the lowest value. So what they've worked out. So they're trying to work out a success rate. And it's around 18 to 23, whereas here it's 3 to 56. So it's a really big difference. Why have they got such a big difference for this one? And why do they have a smaller difference here? What do you guys think? Larger IVF treatments, it is to do with that. Smaller sample, yes, good. It's like if you were to roll a dice 10 times and count how many sixes you have, there'll be... Okay, no, let's say we roll a dice 6 times. You're expected to get a 6 one time, right? But you might not. You might get it 0 times. You might get it 3 times if you're lucky. But if you were to roll it 100 times, if you were to roll it 60 times, so let's say we roll a dice 6 times, you're expected to get 1. You might get 1, you might get 0, you might get 3. But then if you were to roll it 60 times, how many sixes would you expect to get? You would expect to get 10 sixes. And then if you did 600 times, you would expect it to be 100. The more times you do it, the more times you roll, the closer you're going to get to your actual predicted value. Because what happens is the results become more, we call it repeatable. That's why when you do experiments, you repeat the experiment to check if it's repeatable. If you do an experiment plenty of times and you keep getting the same results, it makes the results more reliable, isn't it? So yeah, if you don't understand this bit, don't worry. That's just an example. Basically, if you do it more times, you're going to get more reliable results. If you do an experiment more times, it becomes more reliable. Okay. So to get the marks, you can say there's a larger number of treatments. So results are more repeatable and reliable. Okay. Which clinic has larger treatments? This one. It's got 1004. Clinic R. Clinic S only has 98. You can't say more accurate. No. More accurate means the value is closer to the true value that it should be. This is talking about range, so it's not to do with accuracy. So you can't say more accurate. Okay. Do you have a blank document? Yeah, so what I do, whenever I upload the YouTube videos, guys, I put a blank document up. So all the questions, I upload it in the comments. So you click on the comments, there'll be a link to the blank document so you can print it out and you can do the question while you're watching the video.